Hello, everyone. Thank you for joining us for today's webinar, A Robust and Scalable Suspension Platform for AAV Manufacturing. I'm Leslie Bepko, your host from Cell and Gene and Life Science Connect. With me today, I have Samir Acharya, Associate Director of Process Development from Andelyn Biosciences. Over the next 45 minutes, he'll be discussing demands for AAV-based gene therapy and manufacturing options and strategies for scaling up and downstream purification. After the presentation, we'll be holding a live Q&A session. Feel free to submit questions as we go along. We'll get to as many as possible during the Q&A, and any that we can't get to, we'll follow up on after the event. If you run into any issues, please try refreshing your browser first. If you need additional support, submit a question via the Q&A. Finally, the webinar will be available for on-demand viewing. You'll receive a link via email as soon as that's available. And with all that said, I'm going to turn it over to Samir to get us started.
Hello, everyone. First of all, thank you for this opportunity to share our work. As Leslie mentioned, I work for Andelyn Biosciences. I'm working in the process development department, and I'm the associate director of the group. Today, I will talk to you about our suspension platform for AAV manufacturing. And in this talk, the broad topics that I'll cover is, one, what is the demand or need for gene therapy using AAV manufacturing? Second, how did we develop a platform to satisfy this need or demand? And then I'll also showcase some case studies, which is client work, which have used our platform for scale up of their gene of interest in our group. So before I go into the description of our work, I'll just tell you very briefly about our company. So we are, as most of you know, cell and gene therapy CDMO. We have a very long history of AAV production. We have a long history because we've been linked to Nationwide Children's Hospital for more than 20 years. And we have made several clinical batches, as indicated on the right side of the slide. In 2020, we spun out of Nationwide Children's Hospital and formed an independent company, which we now call it Andelyn Biosciences. In addition to Nationwide Children's Hospital, we have established partnerships and strategic investments by Cytiva and Paul, which is part of Cytiva now. We are based in Columbus. There are 3 facilities in Columbus, within few miles of each other. The Andelyn Plasmid Center, whose job is to make research-grade and GMP plasmids. The Andelyn Development Center, that's where we are housed. In addition to process development, we have the preclinical manufacturing division called the Viral Vector Core and the Analytical Development Division. The third is the Andelyn Corporate Center. That is the primary site for all clinical manufacturing productions for all phases. We are constantly growing, and we have more than 200 employees, and we're excited to be part of this journey. So now let's go to why gene therapy and what is the demand for gene therapy. So one of the drivers of using gene therapy as a mode for curing disease is the fundamental discoveries that happen in the labs all over the world, which then result in an idea being formed, can we use this for curing patients? And that's our main focus anyway. So the biggest driver for going ahead with the gene therapy as a tool for patients is manufacturing. That drives the number of clinical cases that are there, which are then decide on which is the phase 1, phase 2, or phase 3. And the success of those clinical trials is what decides what will be approved and what will not be approved as a drug for curing these diseases. So on the left side, this slide is adapted from a paper which was published last year. And it pretty much simply highlights how the demand for gene therapy has increased. This is specific for AAV-mediated gene therapy. As you can see on the left panel, It highlights the percentage of clinical trials which are in phase 1, phase 2, and phase 3. And over 20 years, you can see how these percentages of trials have increased. If you just go to Google and type in number of clinical trials today on the National Cancer Institute website, you'll see more than 200 clinical trials already being done. So while the clinical trials is a very essential aspect of a drug being made, the success of this clinical trial is key for the next stage, which is FDA approval. So on the right side is a prediction of, in the next 5 years, how many clinical trials will go to completion. Now, completion of clinical trials is dependent upon, very clearly, on how the product is manufactured, how is the quality and the production capacity of those manufacturing divisions. So as we can see, that there is a constant expectation of clinical trials being completed. And once the clinical trial is completed, there is clearly an expectation of an FDA approval to come for at least some of these trials. So starting from 2017, where the first AAV-based gene therapy was approved, it was Luxturna and Zolgensma. This year itself, as you see in the bottom left of the slide, we have 2 more drugs, AAV-based drugs, which have been approved. This has got, so from 2017 to now, the number of approved AAV-based gene therapy drugs are 5, and we expect them to grow as more and more clinical trials get completed. So in addition to this, the next slide then shows, it just highlights how many trials are in phase 1, phase 2, and now, which as expected is the majority of the trials, but there are significant increase in the phase 3 trials on the leftmost panel, and some of them are going to phase 4. When one looks at what kind of serotypes are involved in these trials, it's a whole range of AAV serotypes, not just the normal ones that we know of like AAV2, 9, 8, and so on, but there are many engineered serotypes which many of the labs are focused on, which increase the efficacy of the drug delivery. So this then highlights how important these serotypes are. And when we look at the rightmost panel shows what kind of diseases they're targeting, is the whole range. And this goes hand in hand with the success in the laboratories, where we understand more and more about the mechanisms of disease. That leads to the initial idea, and then the manufacturing then takes over and decides what kind of diseases we can cure using gene therapy and using AAV as a mode of gene therapy. So one of the critical aspects of a clinical trial is the amount of dose that is given in these clinical trials. And so the next slide then shows what kind of doses are being used in these clinical trials. So when we talk about dose, it's in a simple way, we say how much of vector genomes is administered to a patient. And when you look at that, we see the left 2 panels show the range of doses that are being used in clinical trials. They come under 2 categories. The top one shows the high dose category, which goes from E15, mid E13 to more than E17 vector genomes per patient. And the dose is more because of how it's being administered to the patient. The top one shows, obviously, it's a systemic injections to the patient. But if there is a targeted injection that is shown in the lower panel, then the doses are a little less, going from mid E9 to E15 range. So this then dictates what a CDMO like us needs to address in order to make a suspension platform. It lays down the case for what is the task ahead of us. Briefly put, we have to develop a gene therapy process or processes that deliver not just high doses, but high quality of these doses, because we want the trials to be successful in order to reach the patient finally. But also, a platform should be capable of producing a variety of serotypes, because that's where the challenges are. So how did we address this? So before we start on how we address this, it's very important to remember that part of making high doses is the scaled up equipment that we have for scaling up. So on the left panel, we show the kind of equipment we acquired to develop this platform. That is in the research division. So for research and toxicology productions, of course, we have the flasks. We have a 50-liter bioreactor, STR50. We have a 200-liter bioreactor, STR200. And for smaller scale-up, we also have the Amber 250M, which can accommodate 8 vessels of 250 milliliters each. And we can use that for, it's a mini bioreactor, and we can use that to optimize our productions. On the clinical manufacturing side, we have all these bioreactors like STR50, STR200, but also 500-liter bioreactor called STR500 and a 2,000-liter bioreactor, STR2000. This then defines our capacity. So if need be, we can increase the production size. So when we go into optimizing production or suspension platform, we are not the only department that participate in that. So this just briefly highlights the different departments that helps us make a successful suspension platform. The plasmid department, of course, is responsible for plasmid construction and plasmid production, both GMP and non-GMP plasmids, and also purification of plasmid, taking accurate care for the quality of the plasmid. Once that is done, the process development group from which I am part of, we decide together with the conversation with the client, what's the dose necessary for their particular gene of interest, which then dictates what choice of cells and platform to use, whether it's adherent or suspension. And then we start making strategies for maximizing viral titers, because that directly relates to the dose. That's upstream development. In the downstream development, we devise purification strategies, which then leads to determining the quality of the virus produced at every stage. Now, together with monitoring titers, we also do excessive in-process characterization for each stage of our development. This ensures that we track the quality of the product coming out, and it also helps in final stages where formulation has to be devised for the final fill of the virus. This then also helps us scale up the process. So because we have immense in-process characterization, it helps us define what are the critical process parameters, the critical quality attributes of the virus that we have produced. Once we have optimized the production and scaled it up, then of course we are dependent upon other analytical tools that analytical development always helps us. They are the ones responsible for release assays, and they help define how a virus is at every stage. Once that is done, then we have a MSNT group, which helps in technology transfer to the preclinical manufacturing divisions and the clinical manufacturing divisions, which is mentioned as the viral vector core and the ACC. So how did we develop this platform? So our goal has always been, in order to meet the demands of AAV manufacturing, we always focused on two aspects of a virus. One is the productivity, as measured by titers, and also the quality of the virus, as measured by a variety of assays, some of which I'll mention in the start. So there are two ways of addressing that. In order to have an as close understanding of how the virus is produced, we have to evaluate the factors as early as when it's in a small cell stage. So some of the factors that we consider for getting high titers or productivity are cellular factors like cell density, cell passage number, DNA factors, including DNA concentration to use, DNA ratios to use, media factors, which transfection reagent to use, what day of the cells should we harvest the cells, and so on. So we can assess these factors one at a time and see how they affect titers, but that's a very time-consuming process, and also it does not give as much interpretation as possible. So we have utilized a strategy which is now commonly being used by several groups. This is the DOE, or Design of Experiment strategy. This strategy it's a multifactorial strategy which allows us to evaluate multiple factors at the same time. And it also helps us evaluate how these factors interact with each other. So if cellular factors are interfering with the media factors, we know it. So an outcome of this study is shown in the graph on the right. So if we have two factors, it can be cellular factor or DNA factor. it helps us define what are the ranges of these factors that we need to work under in order to maximize the titer. And the color red is the region which has, at least in this graph, shows that's where we need to be. So it shows that we need to be at a lower range of factor 1 and the intermediate range of factor 2 to get maximize the titer. So likewise, most of these studies are done in a small flask, and then like the upstream development, we also do downstream development, and I'll talk to you about some of these factors which are involved in successfully developing a downstream development strategy. So the next slide then shows an example of a DOE study that we used to understand how Factorweed, which is a transfection reagent from Poly Plus, helps increase viral titers. So the left panel shows 3 such factors that we evaluated. One is the transfection cell density, other is the factor V to DNA ratio, and the third is the total amount of DNA per million cells that we use. So what is shown here are contour plots, and blue is low titer, red is high titer, and this gives us an idea of what ranges of each of these factors to work with in order to maximize titers. So a series of such experiments then leads to us defining the ranges of every factor and what you should work with to maximize the titer. The middle panel this shows a similar graph as you showed in the last slide. It's called a response surface plot, and it shows when we compare cell density and DNA concentration, what is the range to use for getting higher titers. A series of such experiments with as many factors as possible then results in a design space. Design space is shown in the rightmost panel, which essentially shows your chances of success. A green area is the one where we have maximum chance of success or the lowest probability of failure, as you can interpret that. For each set of factors, we have these green zones and the red zones. That then is the design space where we want to be in order to have an optimized production. So once we have done this, we start the process of scaling up. So before we start talking about scale up, I'll just give you an example of how we use this Factorweed-based transfection reagent, or Factorweed transfection reagent, to evaluate how different serotypes titers of different serotypes are affected in this. So as this slide shows, we evaluated the titers using Factorweed and compared it with another transfection reagent, which is polyethylenimine. That's a historical reagent that we were using. And as you can see, for 2 different medias, Factorweed worked much better than the historical PEI-based transfection for giving us high titers. In some cases, like serotype A, which is in the left panel, we got as much as 10-fold increase in titers at harvest. And for some serotypes, like serotype B, we got about 1.5 to 2-fold increase in titer. So these are the kind of discussions we have for clients, and that's when we are deciding which transfection reagent to use. These are the kind of experiments which help us evaluate not just which reagent will give you the best titer, but also from the cost perspective, because economically we have to be cognizant of the fact of how much it's worth to have that. So once we have done this small scale characterizations, this is typically done in 125 ml flasks, so we can evaluate as many factors with much less cost input and get much out of the information that is needed for scale up. So once we optimize these conditions, then we are ready for a scale up. And once we decide to scale up from small flasks to big flasks to bioreactors, we evaluate, we have the same strategy, DOE strategy, but a different set of factors come into play. For example, we start evaluating what are the transfection reagent complexation parameters. For example, as we scale up, the mixing of the transfection reagent will be one factor because the volumes of mixing increases. The complexation volume is important. What percentage of the buffer will be used for complexation? What is the incubation time? And so on and so forth. So just like the previous slide, we evaluate these parameters together with the DOE strategy. We evaluate mixing parameters. We evaluate bioreactor specific parameters. For example, what is the impeller speeds to use? What is the power input? What is the gassing strategy? And because that's very different in those compared to flasks. once we have optimized those parameters, then we are ready for assessing how good our optimization is and can we really scale up, or do we have to work more? Since we are talking of scale up, a very important component is downstream development. So once the cells are harvested, we have to remove those cells. That's clarification. We take the option of concentration, which is TFF, or ultrafiltration and diafiltration. There are many parameters to be considered there: what kind of filters, what size, capacity of filters. Likewise, we evaluate many column chromatographies, and so on and so forth. So a very important component over here is in-process analytics. Remember, I mentioned that we always have not just productivity in mind, but also quality of the product coming out at each stage. And for that, the more analytics one has, the better one is to understanding how the system is working. Can we get control of each step, and how do we control that? That's our main goal here. So some of the in-process analytics I mentioned here, like can we assess the translation complex size? Is there consistency in size of the complex as we go from small scale or the large scale? If there is consistency in size, then we can see if that has an effect on titers, or can it lead to consistent titers? So the Bindle panel, of course, it shows you such a study with that, but the rightmost panel then is a DLS measurement, which measures the sizes of transcription complexes. What is shown here is the sizing of a 50-liter transfection mix with DNA. And what we see is a unique size, and the size distribution is unique, not many peaks, which will show many different sizes. And we have optimized this. So this was after optimization of the complexation volume, incubation time, and we saw the same size as we went from small scale to larger scale bioreactors. That then shows and gives us confidence that if we control this, we'll have a significant transfection efficiency, and that's the first step in giving us consistency in titers. So what is shown in the panel below is how do we use analytics to select for membranes, whether it's in this case, we are showing TFF membrane screening. So we have developed an HPLC-based method which can scan through the different protein profiles that come out of different TFF membranes. And in this panel, we have shown 5 different membranes, and we just compare the profiles. As you can see, the outlier in this is the blue tracing. So we can clearly say, okay, we can use the other membranes, but not that. And in this day and time where supply chain demands are important, this becomes a critical aspect, because we do want to have alternate membranes or alternate methods for each step, so that supply chain is not an issue, rate limiting step for production. So we use these, and then the next slide then shows the results of such an optimization. So once we optimize the factors at a small scale, we optimize at a larger scale, then we went about testing how our process compares in different aspects of production. So as you can see in the leftmost panel, the harvest titers at a 125 ml flask or 50-liter flask are consistent between 3 to 4E11. This is a serotype E, and I'm going to talk about serotype E a lot in this presentation. So the middle panel shows what are the recoveries of this serotype, and as you can see, whether it's 1 liter, 2 liter, or 50 liters, the percentage recoveries are in the similar range from 85 to 100%. Likewise, the TFF recovery is also consistent, showing that we had selected the right TFF membrane. Our recoveries are consistent whether we do a small scale or large scale. We have gone on to develop or measure these for 200-liter scale, but at this stage, we were keen on understanding how our process will work in a 500-liter or a 2,000-liter bioreactor. For this, I'll present you a case study which we collaborated with the Paul Process Development Services, who had the 500-liter STR 500 bioreactor and the STR 2000 bioreactor. So we use these 2 bioreactors to see if our optimization works at these 2 scales. We use a 500 liter for the STR 500 production, and we used 780 liters. Because of supply chain issues, we could not scale up to 2,000 liters. But regardless, we used the STR 2000 bioreactor to assess how our process will work in this scaled up platform. So our main strategy was just to evaluate growth parameters, compare the growth parameters in the STR50 or small flask with what the growth is in the STR500 and STR2000. We also compared the productivity, and as for each of these bioreactors, we always have a standard control for a small scale. So in case there's an issue with the small scale flask, we can then assess it's not the bioreactor, it's the small scale flask which is causing an issue. So the next slide then shows us the growth and metabolic profiles across scales. So as you can see, the different color tracings correspond to the 50 liters, 500 liters, a 500 liter control flask. We are talking about 1,000 and 1,000 control flask because we are, instead of writing 780, we did 1,000. As you can see, the cell viability profiles across different days post-seeding are similar for all of them. Most of the profiles are similar even for total cell density per day, pH variations, and the glucose consumption per day. There is a small difference that we see in the orange line, which is the control flask for STR2000, and the purple line is the STR2000. So for each of these different, there's a small difference, and I'll come back to that because that has a significance in what we saw in the final titers. So this then slide shows us what are the titers of this. So again, serotype E was used, and we had an idea that expected titers in serotype E were between 3 to 5. And for STR500, we were right on the money. You can getting around 5 or 5.3 vector genomes per mil as a titer. And STR2000 gave the lower end of the expected titer, which is 3 vector genomes per mil. To evaluate whether it was the STR2000 or something else, we did the control study shown in the right side, and we showed that in the control flask, the small flask, we got lesser titers even there. So there's roughly a 1.5 to 2-fold reduction in titer there. So this led us to believe that the reason why we have a lower titer in the bioreactor is not because of the bioreactor conditions, but because of the control flask. So which can be a variety of reasons, and I'll not go into those now, but we have investigated those. So when we combine this with overlay this STR500 and STR2000 with our internal development work, the middle panel then shows how this is varying. So whether we go for 125 ml flask, mini bioreactor, 2 liter, STR50, STR200, and so on, this pretty much shows consistency in scale up between 3 to 5. E11 was what we expected, and that's what we got. We've also done this scale up with serotype D. What is shown here is scale up from 125 ml flask to STR50, and we can say for this serotype, again, there is consistency in titers. We get around 1.5 to 2E11 vector genomes per mil. So this then shows us that we have consistency in our titers, and that kind of reinforces that our optimization, which was based on a DOE, is robust. So the next stage following the optimization of harvest titers, the next stage is purification. So we have developed 2 different modes of purification. One is the classical ultracentrifugation method, and which is shown on the left side, and the right side it shows the all-column chromatography method. The main difference is where we remove the empty capsids to form full capsids. In the ultracentrifugation method, we harvest the samples, clarify the harvest, concentrate by TFF, and then go into separation of empty and fulls using an adexnol-based gradient centrifugation. Once that is done, we extract the full capsids because it bands very clearly in a region we know, and then we concentrate the virus full capsid with the ion exchange chromatography column, and then followed by concentration and final fill. In the all-column method, the steps are similar, except that after TFF, we have a capture chromatography step. This capture chromatography steps will enrich for the AAV, and then the empty to full separation is another column chromatography, which separates the empties from fulls, and the rest of the steps are similar. So we have tried a variety of capture steps. It can be ion exchange or affinity chromatography that we have used, which can definitely capture the specific capsids. Likewise, we have 3 to 4 empty to full separation chemistries, depending on the viral titer. That's where the challenges are. How can we separate those serotypes with the empty to full chromatographic columns? And we'll give you some examples of that. So we have developed these two methods. And so the next slide will show what are the recoveries at the clarification and TFF stages. And then the other slide will show how does the ultrasimplification method compare with the all-column method as far as enrichment of full capsids are concerned and the purity of the virus that we have obtained. So this slide shows then our clarification recoveries. And as we go from 1 liter to higher scales, we see a consistency in recoveries. The first 4 bars over here show is an average of 4 different serotypes. So, which is important to know because our platform is not serotype dependent as far as the clarification and TFF is concerned. The STR500 and STR200 was done with 1 serotype for obvious reasons. It cost a lot to do this. But again, we have optimized clarification and TFF such that they show consistent recoveries across scales and across serotypes, which is good to know, and it shows the robustness of our process. This then slide shows you for 2 different serotypes, what are our final recovery? When we compare to harvest, what is the final percentage recovery of a final purified AAV? And both for serotype D and serotype E in the ultrasimplification method, we get around average of 30 to 35% recovery. The purity has always been more than 99.5%, and we have confirmed that with host cell protein assays to confirm that our purities are really high, which then further strengthens the quality of our virus being produced. The bottom panel then shows how we developed an all-column method for serotype E, and we have used 2 different vector transfection reagents, PEI-based or vector-wheel-based transfection, and this slide shows 3 different bioreactor studies that we did, and we see the recovery between 30 to 60% from each of these steps. More importantly, when we send our final viruses, purified viruses for AUC measurement, the analytical ultrasimplification method for determining what are the percentage of full capsids, which is by far the most important attribute that we all in the field care about, we got more than 90% purified full capsids. The ultrasimplification method, which is the historical method we always use, not just for suspension platform, but also for adherent platform, has consistently shown more than 90% purified full caps. The purity, this is just an example of the SDS gel that we ran. It's a Cipro orange stain gel that a QC or AD departments run. And whether you have serotype D or serotype E in the first method, ultrasimplification method, or the all-color method, the purity is similar. We have assessed this not just by SDS gels, but in the in-process development in our groups. We've also used capillary electrophoresis as a routine measure for quantifying purities. So, like I mentioned, quality of the product coming out at each stage has always been important to us. And in order to measure quality, we have to develop methods to measure that quality. So one is titers, other is empty to full ratios. So we routinely send our viruses outside for empty to full determination from using analytical ultrasimplification, but that takes 2 months. So we decided to develop in-house HPLC-based methods to resolve empty to full. So for serotype E, we have developed an ion exchange method for resolving empty to full capsids. In addition, we have developed size exclusion chromatography methods in HPLC for impurity profiling, as I showed in one of the earlier slides where we were screening for TFM membranes, but also for capsid quantitation for serotype E and other serotypes. We have done that. We've also developed a reverse phase method for tracking how the impurities get cleared across the process, because finally, even though we have more than 99.5% pure viruses based on a protein estimation, but we want to make sure that other impurities, like if you're using idexanol or chloroquine or phenol red, do we get them removed? And we have tracked this across our purification platform. What is shown here is an example of using such an the size exclusion chromatography for showing how the impurities get cleared. The orange line is the prior to TFF step. As we can see, there are the virus comes out in the very early on, within 10 to 12 minutes. That's where the capsid peak is. The protein impurities come out at 16 to 20 minutes, and beyond 20 minutes are total permeation impurities. So we can see that following TFF, which is the green tracing, we hardly see the other impurities. And this slide then shows us how we have used the HPLC-based method to quantify where we are separating the empties and fulls. So on the right side is the scheme of how we purified the viruses. When we come to the capture step, our viruses have primarily both empty and full. There are two peaks shown on the left side. But when we go through the empty-full separation chromatography, then we see that we primarily get full capsids. And the ratio of these capsids from the HPLC method agrees with what we have seen in AUC methods. So both methods report 88 to 98% full capsids. And this shows us that we have a good method which we've established for at least serotype E. And we always talk to clients, and if they want development, we can develop such methods for different serotypes. Just as a reminder, each serotype, each GOI will have slight differences in how they separate in the HPLC-based method as well as in the empty full separation chromatography. But that's what we work in the development side of things. So at this stage, since we developed a whole platform, we decided to do another case study with another client, and they were also working with serotype E. So we thought this was a good opportunity to assess the development. Now we have developed our platform based on a DOE, but it's not necessary to do a DOE. We can do a yield assessment to see the condition that we have optimized, are those conditions sufficient for the gene of interest that the client is using. So in this case, the first goal was to evaluate serotype E in a small scale for yield assessment using our optimized conditions and evaluate whether we need to go for DOE or not. So the next step was if we do a scale up and optimize purification at the 2-liter scale, and that's where we decide if that works, that development, then we'll go for the 50 liters production, which the client needed for their animal studies. So the next slide then shows how we obtained, it's a summary of all the productions that we did. So the leftmost panel shows us the small 62.5 mil flask study, which showed that our optimized conditions were sufficient to give them titers that they were content with. So there was no need for optimization further. We used that to go to the 2-liter scale. And again, we got 311 vector genomes per mil. We did the purification using, in this case, the client decided to choose an idexanol-based purification, and that's what we can do. And they got satisfactory yields and purities. So then they decided to go with the 50-liter scale, and this graphs over here show how the titers were consistent across scales. Left panel shows going from 62.5 mil to 2-liter to 50-liter. We got between 3.5 to 411 vector genomes per mil at the harvest titer. The final yields are in the middle panel, and we expect the yields going from E13 to E15 at the 2-liter scale to E16 final purified vector at the 50-liter scale. So when you compare the yield at harvest to final yield, we get around 30% enrichment of the virus at the end, and that's what we have observed with serotype E with the ultracentrifugation method. The right panel then shows the gel, and this met all the criteria necessary for purification, and we completed the study for that. So this then shows us that we have developed a robust scalable platform. We have our own conditions with this cell line. This was a commercial cell line that we used. this is suitable for all phases. It consistently gives high yields, high recoveries, and the purity and quality across scales. Now, our platform is developed based on DOE strategies. We have a program which is a stepwise progression during scale-up, and we talk to the clients to develop that, and that's where the discussion then decides what steps to do, what steps we can skip. Our goal is always to ensure quality and yield. Now, one of the things which is very important as we go from development side to the manufacturing side is is there equivalency in equipment and materials. So we use the same equipment in non-GMP and GMP facilities. We use the same materials in non-GMP to GMP. So the same platform process goes from R&D side to GMP operations. That then assures that there's no need for re-establishing equivalency of alternatives, because we have done that in the development side of departments. We have the same analytical assays, so there's no ambiguity about the different analytical assays, which may lead to some kind of different comparisons. So we have the same assays, we have the same QMS system in our company. So we are confident that once we do a development optimization at the development side of department, we'll be ready for clinical productions or preclinical productions. Since we had developed a platform using a commercial cell line, we also decided to develop our own cell line. So we have developed our own suspension cell line, and that that's shown over here. We adapted an adherent cell line, which we had, into a suspension adapted serum-free cell line. It's called SASF cell line. This cell line was clonal, and we established clonality using the WIPS machine, which is shown over here. Essentially, we seed single cells in a 96-well plate, and this monitors and captures data which shows that there are single cells in the green cells, and the red cells show they're not single cells. We go ahead with the green cells and then further develop these different cell lines, clonal cell lines, and decide which one is suitable for further development. So likewise, in this fashion, we have isolated a single cell clonal cell line, which is giving high titers with AAV using our methods, optimized methods. And we've gone ahead and made a GMP cell line, master cell bank from this, and that has been tested and is ready for production for plant gene therapies. So the next slide just gives you an example of how we are using our own optimization with this cell line. And so far, with 2 different optimization rounds, we see that for serotype D, our cell line is working better than the commercial cell line, which is in the middle panel. And we have also now beginning to use in the STR50 bioreactor. So this cell line can be used for client productions, and we already have some client productions going on with this cell line. So in summary, we are a concept to commercialization CDMO. We are one team, end-to-end partner. We can partner with you to go from starting materials, whether it's plasmid manufacturing, vector design, cloning, or cell line development, go into development that we talked about. Process characterization studies can be done, then go directly into manufacturing, and then going to fill and finish. In addition to the suspension cell line, which has developed in the last 5 years, we already have more than 20 years experience in an adherent platform. All these different aspects are controlled by our analytical development and quality control departments. Of course, we all have an oversight with quality assurance, and we also provide regulatory support for all clients as you go into IND filing. So we are open for flexible partnership models, and we have different departments which will help you make an effective partnership. So this is where I'll end, and thank you for your attention, and I'm so open to questions.
All right, Samir, thank you so much. Great presentation. And I know you were a little worried about your timing, but it was perfect. I mean, you're spot on. So great job there. I'm going to give you a little bit of time here. So take a look at the question that did come in there. If you haven't had a chance to submit your question yet, please do so. We are going to get to those. Starting off here, Samir, besides titer, can one monitor different viral attributes at the DOE stage to assess quality of the virus?
That's a great question. And I think this goes to the heart of quality, right? So I said that titer is one aspect, how good it is. So the more assays one has, which are specific for a harvest stage, we can assess that. Some of the assays that are currently available are, let's say, capture ratios. So we, in a DOE platform, we have a variety of conditions which will give high titers, but not all conditions will give you a high capture ratio. So some of them will have more empties than others. So if you have a titer like an affinity chromatography ELISA assay, then we can definitely do that, and we have done that. Likewise, if there are assays which are quantifying, let's say, vector integrity, we can definitely do that. So the best condition will be one which has the entire gene of interest in the virus. And the more assays we have which measure that, and we have some assays which do measure that, we can then say, okay, these conditions worked well with titers, these conditions work with maximizing full to empty ratios, and these also conditions work with maximizing vector integrity. So yes, as more and more assays get developed, we are able to do that, and we have done that with some clients. The challenges in developing assays at the harvest stage is because obviously it is it's a mix of everything at the harvest stage. So the answer to the question is yes, we can do that. Great question.
All right.
There's a question. Sorry.
Okay. I wasn't sure if you had something to add on to that. Are you ready to move on to the next one?
Yes.
Okay. How about this one? During purification, if the column chemistries do not allow for separation of empty from fulls to satisfaction, how will you proceed?
So this question really addresses the heart of challenge in all column-based chromatographies. So the capture column can work. So far, our capture columns have given more than 90% purity. But the empty full separation, although we have different chemistries that we try, but with the different engineered capsids coming up, that needs to be worked out. If all the chemistries don't work, in order to save time, we already have ultracentrifugation method. It's not just idexanol. We also have the cesium chloride method, which is a time-tested method. So the purity of the virus coming out of the capture step, we have reached very high levels of purity there. So we can then go straight into the column chemistry if that's available. Otherwise, we can go into the cesium chloride or idexanol-based centrifugations. We're trying to avoid centrifugation because as we scale up, the volumes are a lot. So that adds to the time for production. But yes, we can do a combination of capture chromatography and centrifugation for purifying the virus.
All right. Great. Moving on here. How about this one? Aggregation is an important issue for AAV. How do you assess this, and at what stage of downstream?
So we have used a DLS, dynamic light scattering, to measure size of the viruses there, and that's how we measure aggregation. But one other thing that is needed for that is purity. So we can assess aggregation if it is more than 90% pure virus. And I think so. What we are getting so far is after the capture column chromatography stage, we assess the purity, and if that's clean enough, more than 90%, then we can definitely do a DLS measurement. That's one of the limitations of the DLS assay. We do measure that. There are now new methods which are being developed, which are mass spectrometry-based methods, and that require acquisition of technology. And that we can use that even at the harvest stage because we have an idea of size of viruses, and we can use that to measure roughly empties and fulls there as well, and aggregates. Yeah.
Okay, great. Moving on, can this platform be adapted to different cell lines?
Oh, yes. So we have, so already I showed you some evidence of using two different cell lines. One was the commercial XP293F cell line, other is our cell line. We have tried different cell lines as well. So when a client comes in, we do give the choice. Some clients may have their own cell line that they want to try. So we give choice at the early stages of development at small scale. We can assess our platform. Yeah.
Okay. All right. Let's see. We have a couple of questions rolling in here too. Again, Samir, if you want to look at those from Sanjay and Sean, not sure if those can be addressed live or if we need more info or those are best one-on-one. So I'll give you a chance to look those over before I ask.
Yes, so one of the questions is, have you moved your all column-based purification process to GMP? So we have not yet done a GMP production, but we are in the process of doing it. So for one serotype, it's all there. We are trying to work with partners who are, the goal is to get to a GMP production in the coming months. We have not done it yet, but the process is already there. So the next part of the question is, if yes, what is the highest scale used in GMP with the all column? So I think I answered that question, that we have yet to do the GMP production with the all column, but the highest scale that we have done in the development side is 200 liters, and the projected scale for GMP is 500 liters. We have not done that yet, but that's in the process.
Okay. How about Suresh's question here? What is the purity of your serotypes?
So we have tested a variety of serotypes using GFP as a transgene, and our purification has always been more than 99% so far. Oh. Whether it's ultracentrifugation based method or for all column. Clearly, for all column, it's not as many serotypes because each serotype will have to optimize the empty full chromatography. So that we are working with the clients. But for adductional based, or I think we are very confident about the purities. We have tested at least in the bioreactors up to 200 liters. We've tested the host cell proteins, host cell DNA, and that's all within the acceptable levels. Yeah.
Okay, and the third part of that question, because this is not my area of expertise, the empty vectors percentage.
So for the serotype that we've tested, I showed you serotype D and serotype E. We have got more than 90% full capsid based on AUC, and with serotype E, we have shown that the HPLC method works the same.
Okay, all right, thank you. Moving on. Let's see. How about this one? Is TFF necessary?
Oh, that's a good question. So not really. So if we are doing affinity chromatography, I think we can bypass TFF. That will save a lot of time and money. And I think that's pretty much established. Several CDMOs and other companies do that. And we are also doing that for different clients. Yeah. It's not really necessary. And by this TFF, I'm assuming the question is referring to the TFF following clarification. There is a TFF before the final formulation that can be done, of course. Yeah. Okay.
All right. One other question in there, but I believe that one needs a little more information for it. So up to you if you want to answer online or off. I'll give you a moment to decide.
I'm just reading through the questions. Answer that.
Yeah. So just for the audience here, just a reminder, you will receive an email automatically as soon as this webinar is available for on-demand viewing, usually within a day or two.
Let me answer one more question, which is what are the highest titers that you have achieved with your platform? Is there a large variation by serotype? So that's what we assess at the upstream stage when we are, let's say we're doing DOE. We optimize this to maximize the titer coming out. Now, these are also dependent upon the gene of interest. And for the same serotype, different genes of interest will give you a different titers. Once we have optimized that, then we do see consistency in the scale up. So if one asks what are the highest titers that we have achieved with our platforms, I can talk about GFP. For serotype E, we've got up to 5.3 vector genomes per mil at a harvest stage. And we are getting, depending on the purification strategy, we got like 60% purified with all column and 35% purified with the final yields there. And this is using a commercial cell line. Our cell line is now for the work that we have done, and that's we are opening it to clients to work with us on that. For serotype D, we are getting up to 3E11 vector genomes per mil or 4E11 vector genomes per mil compared to the commercial cell line, which was giving 1E11 vector genomes per mil at harvest.
All right, great. Well, not another question, but I am going to echo this. Eric says, great job, Samir. I'm going to say the same. So to our presenter, thank you so much to everyone for joining us. Thank you. If you'd like to get a hold of Samir, his contact info is on the screen. If you have any other follow-ups, you can contact him through there. Otherwise, we hope you can join us again soon. We look forward to seeing you. Thank you so much for joining us today. And hope to see you all again. So thank you.
Thank you so much. Bye-bye.